All right, so we are now being recorded. And just for the record, I will reiterate, the purpose of this video teleconference is to review, okay, especially for you, the students, on what was done, some principles that were supposed to be observed in the previous exercise. So that will be done in a form of question and answer, okay, as we'll have a self-quiz again later on. And it's also a venue for you to ask questions and to raise your concerns about this course. As such, I really encourage everyone, if you are listening to this via the recording, no, later, I will encourage you that next time you join the video teleconference so that it's better if there are more of you who are joining live. Okay, so I will now start my presentation. So if you notice, all our video teleconferences are actually planned, okay? There's a specific schedule for them. And right now, we are exactly on that day when we are supposed to have a video teleconference at 2 p.m., not 2 to 3. Um, so again, that will be one less reason for you not be able to join the video teleconference. So I really encourage you all to join. So we are now performing exercise 4. We call it exercise 4, but it's actually the other exercise, osmosis and plasma membrane. Not the one reflected in the calendar because I decided in the last minute to switch them because this exercise that we are doing last week, the exercise last week was actually, not last week, this week, osmosis and plasma membrane is actually very related to microscopy and the hemocytometer. Okay? So even more related than proteomics. So we are going to do the original exercise for proteomics next week. So here are the rundown of what we have done so far. Okay? So I'm not gonna ask you anymore one by one, how are you? But you can always message me if you have concerns later on. But here are the things that we have done so far. So you've undergone exercise 1, micropipettes, and hopefully you have learned the important lessons on micropipettes from that exercise. We also did exercise 2, microscopy, with the addition of micrometers. So a majority of this activity was a review for you from your previous biology courses. Okay, so I really hope that you will have confidence in using the microscope, no, and the micrometers if you do it actually. Okay, because you were only able to watch the videos on this. And then freshly from last week, you did the hemocytometer exercise again via video. So I really hope that if you all have the hemocytometer actually in your hands and you are asked to make cell counts of yeast cells, that you will be able to do it based on what you've learned from the videos and online lessons. So right now, this week we are currently in progress of finishing osmosis and the plasma membrane. Okay, so this is also the focus of our self-quiz today because we already had self-quizzes for exercises 1 to 3 the other weeks. Okay, so are you ready for this self-quiz? May I hear from you? Ready? No answer? I don't have, I'm not giving you a choice to say no. Just say you're ready.
Ready, sir.
Yes, sir.
Okay,
Ready kayo, sir.
so prepare maybe your notes there if you have your notes in front of you. Okay, or if you have a calculator because there are some calculations in this exercise. Okay, I hope you've gone through that. No, there's a calculation portion. Please prepare your calculator or your cell phone for your calculator. Okay, so again, this is just to compete, perhaps healthy competition amongst yourselves. And what's even more important is to sort of a benchmark for you so that you'll know if have I really learned something in this exercise or I don't know about that. How come? Okay, so that you may review yourself again later on if there are some things that you have missed. Start with number one. The diffusion of water across membranes is called
Osmosis, sir.
osmosis. Are you sure about that? Any other answers?
No, sir. Back ko sa yung answer ng osmosis, sir.
All right. So if you have viewed our recorded videos in the lecture, no, um, it's also very timely that we're talking about the cell membrane also within this week. I have already uploaded the video on osmosis also, no, in the lecture. So the diffusion of water molecules, so it's special type of molecule. Water molecule is called osmosis. So when we say diffusion, meaning to say the water molecules will flow from less concentration of water molecules. Am I correct? To more concentration. Am I correct in that sense?
Dili, sir.
Dili di ay. Okay, what's the correct answer?
Bali,
Kung,
sir.
kung water, sir, kay unsa?
From higher
From greater concentration. Yes, to
concentration to lower
going to
concentration,
higher
sir.
to lower concentration. Yes, that's correct. So you sort of think like a dam, di ba? Yung dam, the water is trapped on one side, so that's a very high concentration of water. When you open the dam, okay, the water will all fall down in a waterfall to the other side. So from higher concentration to lower concentration, that's osmosis, no? So that's an analogy, no? Not necessarily the dam is osmosis, but we're talking here about cell membranes which are semi-permeable. Okay, so water will naturally diffuse. No, that's diffusion from higher concentration of water to lower concentration of water. What's the term? Down the what? Down the
Concentration gradient, sir.
okay, down the concentration gradient of water. Okay, so you have to be specific of water because this is osmosis. Number two, water primarily passes through blank in the plasma membrane. So where does water pass through?
Una to unsa, sir? Aquaporins.
All right, the aquaporins. Okay, so water cannot just pass through the cell membrane, no, the bilayer. No, water will bounce back. No, there is no way, there is no passageway for it to go through the cell membrane except the channels, no, which are always open, called aquaporin. So these are special channels for water molecules alone. That's correct. Okay, so when water diffuses in or out of the cell membrane, they always pass through the aquaporin. Okay. Number three, a solution that has a higher concentration of solutes than what's in the cytoplasm of the cell.
Kuan, sir. Hypertonic.
Isotonic, sir.
Hyper,
Hypertonic.
iso, hyper, I think seawater, no? Kanang asin kayo nga tubig, no? From the sea. Seawater. What's the correct answer? Hyper, iso.
Si Kyle, sir. Hyper.
Hypertonic. So that is correct. Okay, so if the concentration of solute, we're talking about solute here, meaning to say what's dissolved in the water. So if what's dissolved is sodium chloride, if the concentration of sodium chloride is greater in the solution than what's in the cell cytoplasm, no? You always compare them, then that is a hypertonic solution. Okay? So what's in the cytoplasm is sort of the point of comparison. Then there's a benchmark. What's the concentration in the cytoplasm versus what's outside? If I'm gonna sort of play with the words in the question, a solution that has a lower concentration of water than the cytoplasm of a cell is a what type of solution?
Itong isotonic din, sir.
Okay, isotonic. Are there any other answers? Ang concentration daw of water in the solution is lower than the concentration of water inside the cell.
Kuan, sir. Hypertonic yapon.
Hypertonic. Okay, so which is which? Isotonic or hypertonic? Or is there another answer? Think about the
Dili
dam.
na siya.
Okay, continue.
Dili na siya hypo cancer.
Naapoy
Basta hypo.
hypo. Okay, okay, so tulo ka buok. May hyper, hypo, iso. Okay, thank you for your answers. Think about the dam analogy. Dam na punta balik. Okay? So there is a side nga inside the cell, there's a side outside. In number 4, it says, what's outside or what's in the dam, okay, is lower concentration of water than what's in the cell. In which direction do you think the water will go? Into the cell or out of the cell? Mas lesser man raw yung tubig sa labas ng cell kaysa nasa loob. So in which direction will the water go? Going inside the cell or outside the cell? In number 4.
Outside, sir.
Going outside because there's a
Inside,
lesser
sir.
concentration of water outside. Diba? Diffusion. We're talking about diffusion. So water will go out of the cell will what? What will happen to the cell pag nawala ng tubig?
Mag-shrink, sir.
Okay? Correct? So what type of
Shrink,
solution
sir.
is number 4? So
Kuan,
what
sir.
type of
Hyper.
solution?
Hypertonic.
Hyper. Okay, so hypertonic. The same as number 3. No? So in number 3, I just sort of tricked you. In number 3, I'm using the word solute. What's dissolved in the water? In number 4, I'm using the word water, the solvent itself. No? So please be wary of these terms because the thought of the sentence or the statement may be the same even if the terms are different. Okay, so just take note of that. Next, 5 and 6. What were the specimens that we observed?
Radiodiscolor, sir, and red blood cells.
All right. Will you be specific on which part of the Rio discolor?
The back portion, sir, of the radiodiscolor. Guard cells
Okay.
of radiodiscolor.
All right. So the cells used in Rio discolor were the guard cells, which are more abundant on the back or lower portion of the leaf. No? Okay. And for the red blood cell, what specific red blood cell did we use?
Red blood cells from the ring finger of the non-dominant hand, sir.
From humans. Di ba? O human red blood cell. Because if you can remember, the human red blood cell doesn't have a nucleus, di ba? If you remember in the pictures that I sent to you or that were shown in the video, there were no nuclei in the red blood cells because they're human red blood cells. Okay? That's our sample of animal cell. Because there are animals like reptiles, their red blood cells have nuclei. No? So you have to be specific which red blood cells those are. All right. So here's an event, the pulling away of the cell membrane from a plant cell wall because the plant cell lost water. What is this phenomenon?
Kuan, sir. Plasmolysis. Plasmolysis.
Any other answers? Your classmate said plasmolysis. So are there any other answers? Or do you agree with that?
None, sir. Agree po kay
No other
Karen.
answers. Okay. So correct. Plasmolysis. No? In fact, later in the pictures, I will show you the same picture that I showed in the videos. You will see plasmolysis. Plasmolysis is lethal to the plant cell or to the plant, no? So, pag meron kayong mga tanim, no, mga plantita, plantito kayo, pero you were so busy that you forgot to water the plant for 1 week, no, and the plant needs watering, say for example, every day, the plant will shrivel and it will die, it will wilt. Meaning to say, its plant cells have undergone plasmolysis because the water went out, no, of the cell. So that's lethal to the plant. What about the wrinkling of the cell membrane of an animal cell, this time caused by the loss of water? What's the phenomenon called? What's the phenomenon called? I hope you were able to go through this in your lab report, which is still in progress. Starts with letter C. Have you come across that word, C? C and then letter R. Anyone wants to try? Sana hindi ko na lang sinabi na starts with C. No answer? We're gonna skip this. Okay, we'll skip number 8. Maybe it will dawn to you later what it is. Number 9, what was used to compare or note the sizes of the cells? What was the apparatus used?
Kuan, sir. Eyepiece micrometer.
Okay, so we used the eyepiece micrometer. The microscope that I used was the same microscope in microscopy, no? So I think you already know the calibrations of our eyepiece micrometer in HPO. That was in HPO. So I don't need to talk more about that one. Okay? So we used an eyepiece micrometer, calibrated already. Then a solution of what salt was used in this exercise? Which salt did we use?
Sodium chloride, sir.
Sodium chloride. Okay. If I may ask, what is the molar mass of sodium chloride? Do you remember? Molar mass. The mass of one formula unit?
Kuan, sir. 58.
58.44 grams per
Okay,
mole.
both of you are correct, no? So we'll just round it off 58, no? 58 grams per mole. Okay, so remember that because there's a question later on that will use that data. Okay, so in number 11, I will be showing you pictures of cells. Then you identify in which solution the cells are, you know, which tonicity. So the ones that you mentioned a while ago, isotonic, hypertonic, or hypotonic. Okay, so I will show one picture at a time. Let's start. Letter C. Anyone?
So 0.3, sir. NaCl
Ah,
solution.
you don't have to be very specific. Just say hypertonic, isotonic, or hypotonic. What is 0.3? Hyper ba yan? Hypo or iso?
Hypertonic, sir.
Alright. Hypertonic. And what are the telltale signs? Telltale. Telltale signs. Look at the picture. What cell is
Nag
this?
guard cell, sir, plant.
Yes, okay. Go. Continue. What did you
Mura
observe?
siya nag-pull away ang membrane from the wall.
Okay, so if I may draw here, no? This one is the membrane of the cell. This is one cell. Okay? And, okay, the cell wall, if I may draw in another color, would be this blue outline. That's the cell wall. No? So there is a gap between the cell wall and the cell membrane, which I will highlight in yellow. There's a gap. So as you mentioned a while ago, this phenomenon is called plasmolysis. And plasmolysis will only happen in plant cells in hypertonic solutions. Okay, so very good. Correct. Next, letter A.
Hypertonic, sir.
And again, what's the sign?
Plasmolysis gyapon, sir, nag-pull away.
All right. Now, so there's also plasmolysis here. You can see highlighted in yellow or outlined in yellow. That's the cell membrane. Okay. And if I may highlight or outline in blue, that's the cell wall of the plant cell. So there's a gap, the pulling away of the cell membrane. So very good. Next, letter G. What liquid was placed in the cell here? If you can remember, a while ago, someone mentioned zero point 0.3
Distilled
for
water,
letters.
sir.
All right, this is distilled water. No, so what is distilled water? Hypo, hyper, iso. Meaning it's
Hypo,
pure.
sir.
Okay, pure water yan distilled water, no? So, meaning to say there's a lot of water in it compared to what's in the cell. So, basically what will happen to the water? Go in or go out of the cell?
In the cell, sir.
Okay, it will go into the cell.
Go
And
in,
look
sir.
at this. Yes. Okay, look at the cells here. These are human red blood cells. In the original picture, you can actually compare this with the other cells, and these are bloated, no? And some of them have already burst, lysed, because they cannot contain the influx of water anymore. So this is hypotonic. What about letter D?
Hypertonic, sir.
Okay, what's the sign?
Shrinking sa red blood cells.
All right. So a while ago in number 8, that was the term. It starts with letter C. What do we see here? The shrinking. What's the term for that?
Try lang ko, sir, ha?
Okay.
Crumple, sir? Or
Crumple. Nice try. It starts with letter C and then R, but no, it's not crumple.
R ang
Correcto siya, CR. Dayon, E. I don't know if you've heard the term in
dili
your basic
crenation, sir.
biology
Ay,
course.
nakaingon si Jan, sir.
Unsa man?
Crenation, sir. Crenation, sir.
Kulang. Crenation is kulang.
Cren-
Sounds like you lack one vowel. I will not say what vowel. Isang vowel na lang. A, E, I, O, U. Crenation lacks one vowel. Wala. Sige, maybe you can remember later on. So correcto, no? Hypertonic, the one who mentioned a while ago, because water went out and the cell shrunk for animal cells. No? What about letter E? In which solution would the cells in E most likely be? Hyper, hypo, iso. The cells in E are the same cells from D and G.
Kuan, sir. ISO.
Okay, so very good observation, no? Because it's okay, let me ask you, why did you say iso?
Kay yung shape, sir, kay na-maintain yung disc-like shape. Makita nimo ang mga indent niya.
Yes. No, okay, you can still see the classic human red blood cell shape, sort of thinner at the center and thicker on the edges. Okay, especially if the cell is on its back, no, facing you. And then you can see the cells that are on their sides. No, they don't look circular because they're on their sides. And this is the normal shape of the red blood cell of humans. And that is only possible if the solution surrounding it is isotonic. Now, meaning to say equal in concentration of water and solute both inside and outside of the cell. It's isotonic. What about letter B?
Hypo, sir.
Hypo. Okay. What made you say it's hypotonic?
Tungod sa iyong kuan, sir, no?
Or
Yung cell membrane kay murag bulag kay sila.
Can you see the cell membrane here?
Dili na kaayo.
Yeah, when you say nabulag ang cell membrane from the cell wall, that's hyper, no? That's the letter A, ito, may gap, di ba? Plasmolysis. Letter C is also plasmolysis. Hyper, yan. Okay? You said letter B is hypo. So I'm asking you,
I
or
mean,
anyone
pinandikit
from
na
the
siya sa
group,
cell wall siya, sir.
okay, nidikit na cell wall. What else? Look what's inside. What about the organelles inside the guard cell? If you see the organelle here in letter A, they are all sort of clumped together, no? Clumped together, very
Kanang,
close together.
yes, sir, kanang separated kay sila, sir.
Okay, so there's a gap
Or
between the
scattered bitaw sila, sir.
organelles. They're scattered, no? These are the chloroplasts actually, and they are all scattered. They are using the whole space inside the plant cell as their container, no? Because the cell is in a hypotonic solution. So the cell, the term there starts with letter T. Puno ng tubig yung plant cell.
Kuan, sir. Turgid.
Turgid. Okay. So the cell in letter B is turgid. Okay. And that's the normal state of all plants. That's why you always need to water your plant, di ba? Or if you have flower cuttings, you always soak the other tip in water para matagal malanta yung flower, no? Because water, a lot of water is needed by plant cells to survive para hypotonic ang environment, para turgid. You don't want the plant to be wilted like that. You want it to be erect. So dapat the cells should also be turgid. And last but not the least, letter F. Mention the type of solution. The tonicity.
Hypertonic gapon, sir.
Okay, so if you observe, similar to letter D, there are those crumpling that you mentioned a while ago. No? One of you mentioned crumpling. And then there was a term crenation, pero kulang ng vowel. Do you now remember what that term is?
Crenated, sir. Or
Di rin. It's also not crenated. Mas malayo na sa term. Crenation is the closest one so far. Kulang lang yung isang vowel. A, E, I, O, U. Wala, yun. Okay. Can't remember. Okay. I'm sure you've heard about this term in your other
Kernatus, sir.
biology.
I hope that.
Crenatus, no. Crenation is really how it sounds like. Kulang lang ng vowel. Didn't you have... okay. Baka nandun sa zoology ninyo, you were not talking about tonicity of solutions in zoology? Anyway, okay, that's the past and you need to know what this term is, but I will not reveal it to you just yet. So let's go to number 12. How many percent of sodium chloride is an isotonic salt solution? O, dapat alam nyo to. Isotonic,
0.9, sir.
0.9 in what?
Percent.
All right. So
0.9, sir.
0.9%. Okay. So not 0.9, 0.9%. Okay. So let's go. Let's proceed. What is the molarity of an isotonic sodium chloride solution?
Kuan, sir. 0.5 0.1540 molars.
That is correct. No, okay, so let me just show you how it's solved. No, when we speak of molarity, ano nga yun? Per liter, di ba, ng solution. Okay? And we are talking here about a 0.9%, okay, na salt solution or sodium chloride solution. Okay? So, if it's per liter solution, that's 1,000 ml of water or the solution. Okay? And technically, that's already around 1 kilogram. Diba? Or let me change the kilogram. Let's just say that's 1,000 grams. Okay, that's 1,000 grams of solution. So not water, but solution. If that's 1,000 grams and isotonic is 0.9%, how many grams will be the sodium chloride? What number do you divide by 1,000 times 100 will have a result of 0.9? What number should this be? X divided by 1,000 times 100 equals 0.9. Wala. If you have your calculators, try 9 grams. If I have 9 grams of NaCl, what's 9 divided by 1,000? 0.0009, am I correct?
Yes, sir.
Oh, times 100,
0.9, sir.
which is this one, isotonic. Diba? Okay, that's the percentage. Now, since we are asked the molarity, let's just convert this 9 grams of sodium chloride into moles sodium chloride. Now, if you can remember your chemistry, and you already answered a while ago, that's going to be 58 grams of NaCl, and that's 1 mole of NaCl. Right. So, if you have your calculators, 9 times 1 divided by 58 is, what was the answer a while ago?
1540
0.1540 or 154 mole
per liter.
NaCl per liter. Or you can just say 0.154 molars of NaCl. So that's the correct answer, 0.154 molars. Right, last question. How many percent of sodium chloride is a 0.5 molar sodium chloride solution? Zero point 0.5 molar moles. Let me erase one. This means that there is 0.5 moles of NaCl per 1 liter of this solution, which is aqueous made of water. Okay? So the question is how many percent? What do we do next?
Sir, convert to grams ang mole.
Okay, so we'll be converting this into grams. So 0.5, I will just omit the units. 0.5 times 58 grams ang 1 mole. So what is 0.5 times 58?
Sir, Ay, 58 lang, sir, no? Dili na 58.44. Kuan, sir, 29.
29, sir.
Right,
29,
29.
sir.
Okay, that's correct. So let us put here instead, this is now 29 grams NaCl over 1 liter of solution. Now, since this is water solution, no, aqueous, we can say that the mass of this solution is around 1,000 grams. Right. Tapos 29 ang NaCl. Oh. So you now have 29 divided by 1,000. What's the answer?
0.029.
Okay, 0.029 and then times 100%.
2.9, sir.
2.9? Okay,
0.9.
2.9% NaCl. Okay, so yun pa lang answer. So, a 0.5 molar NaCl solution, aqueous solution, is about, we are estimating 2.9% sodium chloride. Okay, so I hope this brings back memory from your inorganic chem. And I hope you'll be confident in the future in converting molars into percent solution, no? Because this is important, not just in cell mol, but also in physiology and even in medicine. All right. So, congratulations. Okay, we are now done with the self quiz, and I hope there's a revelation for you on what things you should review on. No, for example, crenation. You still don't know what the term is, crenation, or do you already know?
Sir, wala gid, sir, vibe lang. Kung gid akong utok, sir.
Okay, so I'll leave that to you. We'll see in your lab report if it's there.
Saba na lang, sir.
Hindi na ako saba. Okay. Sige, so prelim project, no, I extended the due date for this. So it's due tomorrow, Friday. So I really hope you will be able to pass na your prelim project tomorrow. No, midnight tomorrow. And then on the following day, also midnight, is the deadline of the exercise 4 that you're currently doing right now. No? So I will wait for this so that I can read what that crenation word is. Right. So these are the deadlines. No? Okay, so for now, that's everything. So if you have any concerns, you may voice them out now. Any questions?
Sir, about sa prelim project, sir.
Hmm.
Is it necessary? Kailangan gud magbutang po og procedure sa estimated cell size, sir? Cell density ba to, sir?
What was the main question? The cell density.
Yes, sir.
Yes, so you really need to make, that's the main goal of the prelim project. You will make, no, your own procedure.
Sa estimated
In or-
sir? Cell.
Oh, in order to estimate the cell density.
Okay, sir. Thank you, sir.
And just a reminder, that's going to be future tense, di ba? Because you will still, it's still a plan. It's not past tense.
Okay, sir. Sige, sir. Thank you, sir.
All right. Any other concerns, questions?
All other lab reports, sir, kay past tense, sir.
Sir, sa drawings
Yes.
karon, sir ba?
Oh. All right.
Kanang pila ka sa ang required i-drawing, sir? Sa kuan.
Red blood cell. Human red blood cell.
Oo, sir. Sir.
Was it indicated? If it's not indicated, I think at most 5 will be okay. Okay. The important thing is you will be able to observe what's happening in them. Yun lang naman. So 5 red blood cells will be okay.
Okay, sir. Thank you, sir. Okay rag 3, sir ba? Kay at most man ang 5.
Oh, okay lang ang 3. Okay, so any other
Sige, sir.
concerns?
Thank you.
All right.
ay sir kanang last lang. Sa kada drawing sir kay necessary gid tagaan silang discussion kada isa or kanang description lang sa ubos sa drawing.
A description for the figure, no, because this is a figure. You always put your description for a figure below the figure. That's a reminder. You put a description, and then in your discussion, of course, you talk about them, not necessarily one by one, no, but you talk about them in general, like what's happening in the figures.
Okay sir, thank you
All right,
sir.
so it's not necessary na you will make one paragraph talaga na explanation per picture, no, or per drawing, no, not like that. You can already sort of summarize everything that you want to say on, say, all of the figures in your discussion. Pwedeng ganun. All right. I hope you've checked the course wall just a while ago because I already returned exercise 2. I already uploaded the checked exercise 2 there. So please do check, and you may look at your scores from exercise 2. And if you have any concerns, just message me. So any other concerns for this forum? Okay, so the exercise number 4, which is plasmolysis and the plasma membrane, I think is our last exercise in cell mol na gagamit tayo ng microscope. No? This is the last exercise na microscopy. The next exercise, which is about proteomics, is already a different type of exercise. Hindi na siya microscopy. We're gonna start using the spectrophotometer already. Okay? So I've already recorded videos for that, and I hope you're excited to see what the activity is like because we will be using the spectrophotometer for that. And from then on, no more microscopy, no? So basic man tong microscopy na cell mol. So now we will be using the PCR, the spectrophotometer, the vortex, or the centrifuge. So ganun na tayo na level. Okay, so I hope you will continue having the drive, no, in this course and in the other courses that you are in. Yeah, so that you will have a better performance. Your performance will become better as the semester continues. All right, so if you have other questions, because we are now bound by time, there's just 1 minute left. You may always message me in eLearn for your question. Okay, so again, thank you very much, guys, for actually joining in this video teleconference. And please encourage your classmates who are not here right now to join in the next video teleconference because I think it's really important for you to be able to experience, no, parang self-quiz. It's different than if you just study by yourself. No, because here you can interact, okay? Plus, I think it's fun, diba, to test ourselves what we've learned so far. So thank you very much, guys, for being here, and have a productive day ahead. And you are now all dismissed.
Bye, sir.
Thank you, sir.
Sir.
Bye, sir.
Thank you, sir.
Thank you sir, God bless.